Good morning, student. We will continue the topic protein metabolism, competition number 5.4, and today's subtopic is transamination. Before that, we will see in short about the amino acid pool. So, amino acid pool is nothing but it is the presence of free amino acid in various parts of the body. It may be intracellular or extracellular. So, amino acid pool constitutes 100 grams of free amino acids. It is very small as compared to total body proteins. So, roughly in an young adult, the total weight of body protein is about 12 kg. So, among these amino acid, glutamate and glutamine, these two amino acid constitutes about 50% of amino acid pool, and remaining all essential amino acids constitute only 10% of the amino acid pool. The concentration of intracellular amino acids is always higher than extracellular amino acids. Amino acids enter the cell against the concentration gradient, so therefore it is an active type of transport. Amino acids are not stored in the body. So this figure shows the contribution of amino acid by various causes and utilization of amino acid. So our body proteins daily undergoes breakdown and resynthesis. So it is called as protein turnover. So that protein turnover contributes 40 grams of amino acids, 400 grams of amino acid to the amino acid pool. That means daily 400 grams of proteins are broken down in our body. So free amino acids are formed, and these free amino acids are added to the amino acid pool. And in our diet, average proteins are 50 to 100 grams. So from this amino acid pool, then body utilizes 400 grams of proteins for the synthesis of same proteins that have broken down. Then 30 grams of proteins are utilized for synthesis of special products. Some amount of amino acids are utilized for synthesis of non-essential amino acids. And in fasting and starvation, some amino acids are utilized for synthesis of glucose and ketone bodies. So this is in this way the amino acid pool is maintained. So in fasting state, there is increased utilization of amino acids from amino acid pool for the synthesis of glucose and ketone bodies. Therefore, the synthesis of body protein is decreased. Now, this is the structure of amino acid, all of us know. The catabolism of amino acid takes place in two major steps. The first major step is removal of alpha amino group from amino acid, and this alpha amino group is removed in the form of ammonia. So this is very dangerous step because free ammonia is very much harmful for our brain and central nervous system. Therefore, utmost precautions are taken by the body so that free ammonia should not be remain. That we will see in details later on. And second step is, after removal of ammonia, what remain is carbon, oxygen and hydrogen. So it is called as breakdown of carbon skeleton or keto acids. After removal of amino group, amino acids are called as keto acids. So removal of amino group occurs by these three steps: transamination, deamination and transdeamination. First, coming to our today's topic, transamination. As the name suggests, transfer of amino group. So in this case, in various tissues, the amino group from an amino acid is transferred to a keto acid, and after transfer of amino group to the keto acid, that keto acid forms a new amino acid, and after removal of amino acid from the initial amino acid, initial amino acid is converted into new keto acid. So in this way, the new amino acid and new keto acids are formed during transamination. The enzymes which catalyzes the process of transamination belongs to a group called amino transaminase, belongs to class 2 of enzyme classification. Each reaction requires specific enzymes, so there is no single transaminase enzyme. There are so many transaminase enzymes for every transamination reaction, and all these reactions of transamination requires a coenzyme pyridoxal phosphate. So this is the process of transamination. First, amino acid one have transfer its amino group to pyridoxal phosphate, which is a coenzyme. Now after accepting this amino group, the pyridoxal phosphate is converted into pyridoxamine phosphate, and that amino acid one is converted into keto acid. Now this pyridoxamine phosphate transfer this amino group mostly to the keto acid alpha ketoglutarate. Now after addition of amino group to the alpha ketoglutarate is converted into glutamic acid, and pyridoxamine phosphate is converted back to pyridoxal phosphate. So this is the role of pyridoxal phosphate in transamination. Now we will see two these examples. First example is alanine plus alpha ketoglutarate. So here the amino group from alanine is removed and added to alpha ketoglutarate, and this alpha ketoglutarate is converted into glutamic acid. After removal of amino group from alanine, that alanine is converted into pyruvate. Now in this reaction, the enzyme is ALT, that is alanine transaminase, or SGPT, another name means serum glutamate pyruvate transaminase. Now second example is aspartate plus alpha ketoglutarate. Here again amino group from aspartate is transferred to alpha ketoglutarate, and after addition of amino group to alpha ketoglutarate, it becomes glutamate, and after removal of amino group from aspartate, it becomes a keto acid oxaloacetate. The enzyme is AST, that is aspartate transaminase, or another name is SGOT, serum glutamate oxalate transaminase. Now if you see both this reaction, then what you observe is alpha ketoglutarate is a keto acid which is common acceptor of amino group. So in the body also alpha ketoglutarate act as a acceptor of amino group from various amino acid. So as alpha ketoglutarate is the common acceptor of amino group, the most common amino acid formed during transamination is glutamate. Hence, if we see the concentration of free amino acids in the body, then we found that the concentration of glutamate or glutamic acid is 10 times more than other amino acids. So this is a summary of transamination. Alpha ketoglutarate is the common acceptor of amino group in tissues. So glutamate is the common amino acid formed in the pathway. Every amino acid requires separate enzyme for transamination. This you should remember. And glutamate formed in various tissues. Now it is transferred to liver for deamination. Now what is the significance of transamination? First is synthesis of non-essential amino acids. If you see here, pyruvate is converted into alanine by adding a amino group. Similarly, by adding amino group to oxaloacetate, aspartic acid is formed. So from various keto acids, new non-essential amino acids are synthesized by transamination process. Now second importance is interconversion of amino acids. When it is required, this occurs when there is deficiency of a particular non-essential amino acid and there is availability of any other amino acid. For example, here the concentration of alanine is more, but aspartate concentration is less. So in this case, the transamination reaction occurs in such a way that first, after removal of amino group, that alanine is converted into pyruvic acid. Then pyruvic acid undergoes carboxylation reaction to form oxaloacetate, and then again by transamination reaction, amino group is added to the oxaloacetate to form aspartic acid. So in this way, alanine is converted to aspartate. Now remember, 3 amino acids like lysine, threonine and proline, they does not undergo transamination reaction. Glucocorticoids stimulates the synthesis of transaminase enzymes. These transaminase enzymes then favors gluconeogenesis. That means they divert the keto acids of formed from amino acid for the synthesis of glucose. We have studied transamination. Now we will study deamination reaction and formation of ammonia. Now deamination. What is deamination? Removal of alpha amino group from amino acid in the form of free ammonia is called as deamination. Now the basic difference between transamination and deamination is, in transamination amino group is removed from amino acid, but it is added to new keto acid, so that free ammonia is not formed in transamination. But in deamination, amino group is removed and free ammonia is formed. Amino group is removed. After removal of amino group from that amino acid, the amino acid is converted into keto acid, but that amino group is released as free ammonia. So deamination is of 2 types: oxidative deamination and non-oxidative deamination. First we will see oxidative deamination. Removal of alpha amino group from the amino acid in the form of free ammonia with simultaneous oxidation is called as oxidative deamination. So these are the various types of oxidative deamination. First, oxidative deamination by glutamate dehydrogenase enzyme. Second, oxidative deamination by L-amino acid oxidase enzyme. Third, oxidative deamination by D-amino acid oxidase enzyme. Fourth, oxidative deamination by glutaminase enzyme. And next is oxidative deamination by monoamino oxidase enzyme. So we will see all these 5 types one by one. First is oxidative deamination by glutamate dehydrogenase enzyme. So it is the major deamination process in our body. Glutamate dehydrogenase is the enzyme which is exclusively present in liver. NAD+ is the coenzyme required for this enzyme. Glutamate dehydrogenase is allosterically inhibited by ATP and activated by ADP. That means whenever ATP is present in the body, then deamination is inhibited, and when there is deficiency of ATP, that means ADPs are available, at that time the enzyme is stimulated and deamination process is also stimulated. Now this deamination generally takes place after transamination. In the transamination, we have seen that various amino acids in tissues transfer their amino group to alpha ketoglutarate, and that alpha ketoglutarate is converted into glutamic acid. This glutamic acid is then transported to the liver. Now here this glutamic acid undergoes oxidative deamination by glutamate dehydrogenase reaction. So here glutamic acid or glutamate, here simultaneous oxidation takes place by removal of two hydrogen atoms. Therefore NAD is converted into NADH, and the amino group which is released here in the form of free ammonia. Next oxidative deamination is by L-amino acid oxidase. It is a minor pathway of deamination of L-amino acids. It takes place in all tissues. This L-amino oxidase requires FMN as a coenzyme. Here the L-amino acid, the enzyme L-amino acid oxidase act on it. Again the same mechanism takes place. Two hydrogen atom along with amino group in the form of free ammonia is released. Here the acceptor of hydrogen atom is FMN, which get converted into FMNH2, and the corresponding L-amino acid is converted into corresponding keto acid. Now in this process you have to remember that when this FMNH2 coenzyme is formed, it get converted back to FMN by donating its hydrogen atom to oxygen molecule. So in this process hydrogen peroxide is formed. So hydrogen peroxide is a reactive oxygen species. Reactive oxygen species means when there is incomplete reduction of oxygen molecule takes place, it will give rise to reactive oxygen species. For complete reduction of oxygen molecule, 4 hydrogen atoms are required. But if 1, 2 or 3 hydrogen atoms are added to the oxygen molecule, it is called as incomplete reduction of oxygen molecule. So incomplete reduction of oxygen molecule gives rise to reactive oxygen species. So this is the disadvantage of this mechanism, because hydrogen peroxide is harmful for the cell. But in our cell the enzyme catalase is present which add oxygen with hydrogen peroxide and leads to its complete reduction. It leads to complete reduction to form water molecule. So here again that I have mentioned, it leads to formation of free ammonia in all tissues. This is also a dangerous effect, because by this type of deamination free ammonia is formed in tissue, whereas deamination by glutamate dehydrogenase leads to formation of free ammonia in the liver, where it can be directly converted into urea. But here free ammonia is formed in all tissues by this deamination, but it is a minor type of deamination process. Second is reactive oxygen species, that is hydrogen peroxide is formed. Next type of oxidative deamination is by D-amino acid oxidase. It is also a minor pathway. It takes place in all tissues. By this mechanism or this enzyme, D-amino acid oxidase acts on glycine and D-amino acids. D-amino acids are very less in our body. For this enzyme, the coenzyme required is FAD. So this is the reaction. D-amino acid undergoes oxidative deamination, that is removal of two hydrogen atom and amino group in the form of ammonia to form corresponding keto acid. Two amino acids are accepted by FAD to form FADH2. Again, this FADH2 donates its hydrogen atom to oxygen molecule while it converts back into FAD. Now this hydrogen molecule then forms hydrogen peroxide, which is a reactive oxygen species, and again the catalase enzyme which is present in the cell will convert this hydrogen peroxide into water molecule. So disadvantage of this mechanism is also same as previous mechanism. Again, free ammonia is formed in various tissues. These ammonia may enter into the blood. From blood it may circulate to the brain and central nervous system, and it may damage to the brain. And second disadvantage is formation of reactive oxygen species. Next is oxidative deamination by glutaminase. It is also a minor pathway of deamination. It takes place in the kidney. Here the enzyme glutaminase is present. This glutaminase acts on glutamine and leads to oxidative deamination of glutamine to form glutamic acid and free ammonia. Here the free ammonia is formed in the renal tubules. This free ammonia combines with H plus ions which are present in the filtrate of the renal tubules to form NH4 plus ions, or ammonium ions are formed. So in this way, this ammonia which is formed in the filtrate of renal tubules neutralizes the H plus ions. If these H plus ions are present in that filtrate, they form urine, and the pH of they decreases the pH of urine, and such urine may damage our urinary tract. So actually here the ammonia act as a buffer in the renal tubules. Again here the free ammonia is formed in the kidney, but it is purposefully formed for neutralization of H plus ions. Next is oxidative deamination by monoamine oxidase. It is also a minor pathway. First you should know what is monoamines. Monoamines means when amino group is removed from the amino acid, they are called as monoamines. So monoamines undergoes deamination by various enzymes to form carbonyl skeleton and free ammonia. So these are 5 different ways of oxidative deamination. Next we will see non-oxidative deamination. Removal of alpha group from amino acid in the form of free ammonia without oxidation is called as non-oxidative deamination. It is of 3 types. There are only few amino acids which undergoes this type of deamination, like non-oxidative deamination of serine and threonine, non-oxidative deamination of cysteine, and non-oxidative deamination of histidine. First we will see non-oxidative deamination of serine and threonine. It is also a minor pathway. It requires coenzyme pyridoxal phosphate. Serine undergoes non-oxidative deamination with the help of enzyme serine dehydratase, and the serine is converted into pyruvate. Threonine undergoes non-oxidative deamination by enzyme threonine dehydratase, which forms alpha ketobutyrate and free ammonia. Next, cysteine. Cysteine undergoes non-oxidative deamination by enzyme desulfhydrase to form free ammonia and beta mercaptopyruvate. When the sulfur SH group from that beta mercaptopyruvate is removed, it forms pyruvate. And last is non-oxidative deamination by histidine with the help of enzyme histidase, and histidine is converted into urocanic acid and free ammonia. Again, disadvantage of non-oxidative deamination is free ammonia is formed in all tissues. Now we will see third step of removal of ammonia, that is transdeamination. So it is also a major pathway of removal of amino group from amino acid. Transdeamination in tissues coupled with deamination in liver. So transdeamination is not a single process, it is a combination of transamination and deamination that we have studied. Here all transdeamination contains all types of transamination in the tissue plus oxidative deamination in liver by glutamate dehydrogenase is called as transdeamination. Now see this figure. Here transdeamination means transamination plus oxidative deamination. Amino acids donate their amino group to alpha ketoglutarate and they get converted to keto acids, and that alpha ketoglutarate is converted to glutamic acid in all tissues that we have studied, that is transamination. Now this glutamic acid is then transported to the liver. In the liver, this glutamic acid undergoes oxidative deamination with the help of enzyme glutamate dehydrogenase, which requires coenzyme NAD to form alpha ketoglutarate. So what is transdeamination? Whenever there is a question of transdeamination, then what you have to write is, you have to write down the transamination process that we have studied plus oxidative deamination by glutamate dehydrogenase, so which is the major deamination process, right? So when you combine these two, then it is transdeamination. Now importance of transdeamination: this type of deamination leads to formation of free ammonia only in the liver. It is generally couples with transamination. So in this case, free ammonia is formed only in the liver. Apart from this, remaining all the deamination processes we have seen, free ammonia is formed in various tissues. The advantage of forming ammonia in liver is that in the liver, this ammonia is immediately converted into urea cycle, immediately enter into the urea cycle and converted into urea. So deamination is nothing but removal of amino group. Transdeamination means removal of amino group in liver. So this is about transamination, deamination and transdeamination, that is removal of amino group.